Hello, my name is Valeria Flores. I am a graduate student at the National Autonomous University of Mexico. Today, I'm going to talk about the findings of my bachelor thesis. My work is called Viability, Effectiveness, and Infectivity of Laccaria trichodermophora in Pinus montezumae roots. So, I would like to start mentioning that in Mexico, a considerable percentage of the forest area is lost every year. In order to address this problem, there are national reforestation programs that use seedlings produced in forest nurseries. But despite this, the seedling survival is lower than 5% in later years. The 13% of such mortality is related with the bad plant quality. A way of improving the plant quality is through controlled mycorrhization, by which the plant growth and the nutrition can be enhanced. The Ectomycorrhizal inoculum may come from different resources, but in my work we focused on the metabolic active mycelia, so-called vegetative inoculum. So we propose to use the following parameters to estimate the potential of a vegetative inoculum to be used in reforestation programs. The first one is the infectivity, that refers to the inoculum ability to colonize the roots of a host. The second one is the effectiveness, that is the inoculum ability to promote the growth of its host plant. And finally, the viability, that is the inoculum ability to maintain its genetic, phenotypic, and physiological stability over time. To evaluate those parameters, I used Laccaria trichodermophora, that is an ectomycorrhizal fungal species associated with Pinaceae family members. First and foremost, this fungus has been isolated and the biotechnological process for its large-scale production is underway. I use Pinus montezumae seedlings. This is a Laccaria trichodermophora natural host, and it has great economic importance in the wood and paper industry. So, as objective, we aim to evaluate the viability, effectiveness, and infectivity of a Laccaria trichodermophora vegetative inoculum. The viability was evaluated by encapsulating the mycelia of Laccaria trichodermophora in alginate bits. The effectiveness was evaluated by inoculating Pinus montezumae seedlings with spores, two different vegetative inocula, and mixed sporal mycelium inoculum. And finally, the infectivity was evaluated by inoculating Pinus montezumae seedlings with four Laccaria trichodermophora strains. Then I will present to you our results. The encapsulation in alginate bits keeps it viable up to 3 months at 4 degrees, and at root temperature, the viability decreases as early as the second month. In the effectiveness experiment, we found that the sporal inoculum affects the seedling survival, but the sporal and mixed inocula were the only ones that colonized the roots. Neither the suspended mycelia and the mycelia encapsulated in alginate bits formed mycorrhizal root tips. The sporal, mixed, and suspended mycelium presented the highest root values. However, the results of the quality indexes were inconclusive. Regarding infectivity, we found that there is variability among strains in their ability to colonize pine seedlings, since only one strain successfully colonized the roots. In fact, the 10th strain developed mycorrhizal structure as early as the 5th week. Here, we can observe a developing mantle, and in the 24th week, we can see mycorrhizal root tips in different developmental stages. At the end, we observe a well-developed mantle and a hardy net that colonize the epidermis and the first cortex layer. We even could see sporums primordia. The other strains presented a structure related with ectomycorrhizal association as hyphae on the root surface, dichotomization, and arrest of root hair growth without forming major mycorrhizae. Our results are interesting because the strain that was used in the effectiveness experiment is one of the most productive in liquid media, so strains with high biomass production did not necessarily correspond to high host colonization and capacity to benefit their host. I would like to end my presentation by thanking the Project PAPIT that funded my thesis, also to my advisor, Rodolfo Angeles, and the Laccaria project leaders, the Dr. Roberto Garibay and the Dr. Mauricio Trujillo, as well as the Laccaria team and my lab partners. Thank you for your attention.